Okay, our last talk of the first morning session is a Proceedings paper, and it's going to be given by Siamak.
Good morning, everyone. I'm Siamak, a PhD student at Texas A&M University. This is a joint work by my advisor, Dr. Jianing Chan, who is also here, and Professor Minguan Zhu at University of Texas at Austin. This is a work based on factorization for RNA sequencing count data, and we are going to show that incorporating covariate information is going to improve the chance for biological information discovery. So, as I mentioned, the primary goal of work is extracting groups of genes from gene expression data that show coordinated expression patterns across multiple groups of samples. This problem has been around since microarray technologies, and the primary approach towards this problem has been network-based problem. As an example, one of the most popular one is WGCNA framework, which is based on weighted graph correlation network analysis. So in such network-based methods, first, a correlation network is constructed from gene expression data based on pairwise correlation between different genes. And then using any clustering method, such as, for example, hierarchical clustering, modules of genes are extracted from these networks, and then these gene modules are correlated with external information, such as treatment versus control, to identify how they're going to change with respect to this additional information. These network-based methods also have been applied to RNA sequencing data, but there are some challenges to do so. First challenge is having data normalization. So RNA sequencing data is going to give us a count data, which are usually over-dispersed, and if we are going to apply this network-based methods, we need to do normalization that may cause losing some information. Then for applying clustering, we need to determine the number of clusters in advance, but we don't know that usually. And most importantly, for those covariate information, we don't have any systematic way of incorporating them. And it's based on heuristic methods. What are we going to do in this work is developing a factorization method that is directly applicable to count data from RNA sequencing. So it will address the first normalization challenge. And it has inspiration from Bayesian non-parametric literature, so it's going to automatically infer the number of latent features or clusters from data automatically, and it has a systematic way of addressing incorporation of sample covariates using a regression layer in this method. So all of this is going to be done in a simultaneous manner with a probabilistic model. So let's take a look at our model. This is the graphical representation of our model. We first start by modeling the count data. We assume that there are capital V genes and capital J samples. So the count for gene V in sample J is modeled using a negative binomial distribution. Negative binomial is a popular distribution for modeling the count data from RNA sequencing because it has the over-dispersion property between its variance and its mean. We use the dispersion and probability parameterization. So the dispersion is RVJ and probability parameter is PJ. Here in this model, we can interpret the PJ as the effect of sequencing depth. So it will obviate the need for performing normalization. This can be seen by taking a look at the expectation of the count. There is a term that only depends on the probability parameter, which is indexed by sample J. So there is no need for doing normalization, and we can use the residual term for subsequent modeling. The latent factor representation of model is achieved by using a matrix factorization over the dispersion parameter of this negative binomial distribution. So this will give us two matrices. the matrix phi, which is the factor loading matrix, which tells us what's the association between gene V and latent factor K. And we have also the factor score matrix theta, which tells us how strongly latent factor K is expressed in sample J. So we are hoping for these latent factors to give us the coordinated expression patterns and subsequently biological insight. But how to incorporate covariate information? Assuming that we have a vector of sample covariates xj, which includes capital P covariates. For example, this can be gender information, batch effect information, or any other information. We assume that the there is a gamma regression prior on our factor score matrix elements theta. So in this gamma regression, we have two parameters. One is RK, which we interpret it as the baseline expression for latent factor K. And we also have a regression term in the scale parameter of our gamma distribution, which is exponential of a linear regression. So here we have the covariate coefficients beta, which tells us how the covariate information P is going to affect the expression of latent factor K. So for example, if we have gender and we discover some groups of genes K, we are going to see how this covariate gender is going to affect the expression of our latent factor across different samples. We further assume a sparsity promoting prior over the regression coefficient beta. This is called automatic relevance determination, and it's very popular in machine learning. And in this prior, we assume that the regression coefficients are modeled using zero-mean normal distributions with precision parameters that are common between different genes. So for those factors, covariates that are not important, this precision will be pushed towards zero. The variance will be pushed towards zero, so these non-important covariates will be automatically eliminated from affecting our model. We also have the problem, as I mentioned in the first slide, how to determine the number of latent factors. So what we do here is using a prior which is inspired by gamma process from Bayesian nonparametrics. So this prior will automatically eliminate redundant factors. If we apply, for example, this model to a real data set of TCGA, breast cancer data set, we can see the baseline expression that have an exponential decay. So what we can do in practice is just look at this table, look at this plot, and just discard non-significant latent factors based on this baseline expression. So this will give us a nice property of just deriving the number of latent factors from the data. So these are the summary of model parameters. We have, as I mentioned, RK is the baseline expression for latent factors. PJ is the effect of sequencing depth. We have factor loading phi, which is association between genes and modules. We have theta, which is strength of latent factors in samples. And also we have, finally, we have the regression coefficients, which tells us the impact of covariates on latent factors. So to infer the model parameters, we use Gibbs sampling, but it's challenging because we don't have closed form conditional distributions for two important model parameters. The first one is dispersion parameter of negative binomial distribution, and the second one is the regression coefficients. So to address this issue, we resort to two famous data augmentation techniques. One is called Chinese restaurant table, which is for negative binomial distribution parameter, and the other one is polygamma distribution, which is popular for Bayesian logistic regression. So more details of the Gibbs sampling inference is available in our paper, and you can refer to that if you need more details. The rest of parameters are updated just using simple conjugacy between priors and or likelihood, which is mostly based on Poisson and gamma conjugacies. So to show how this model can perform comparing to network-based models, we apply our model and two other ones to the 3 datasets from TCGA, including breast, lung, and kidney cancers. So to analyze, we first discovered gene modules using these models. Our model, which is DNBFA, Dependent Negative Binomial Factor Analysis, WGCNA, and DIFCOX, which are two popular network-based models. After discovering the modules, we extract the eigengenes. Eigengenes is a kind of representative of the expression of gene modules. It's achieved using principal component analysis. Then, to see how these representatives of gene modules are differentially expressed across treatment and control, we apply a t-test and we achieve the p-values using the t-test. So here we see the sorted negative log of the p-values. As you can see, the module discovered by metric factorization show more significant differential expression compared to the network-based methods. So they are good candidates for giving us a set of genes that show differential coordinated expression patterns, and they may have some biological insights within them. To more thoroughly look at the results, we see the top eigengenes expression patterns for all methods and all 3 data sets. What we can see here is that eigengenes discovered by our model are more consistently differentially expressed across samples. If you look at the red and blue bar plots, you can see that for the first row, there are the variation within the groups are less for us compared to network-based models. And to see how incorporating the covariate information, one of the main properties of our model, can help biological insight, we also applied this model to a dataset of autism. This dataset includes 36 samples from control group and 23 from autism patients, and there are covariate informations available for samples. This covariate information include the site of sample collection, which reflects the batch effects, age, sex, and brain region that the samples were collected from. So what are we going to do? We are going to apply the model once, including all this information, and second, ignoring this covariate information to see how they can change the discovered modules' biological meanings. So to assess the biological meanings of the modules, we use gene ontology enrichment analysis. And here in this plot, we can see the sorted negative logarithm of p-values again. The blue dots are for the factorization model when the covariate information of samples are incorporated, and the red dots are the ones that the covariate information are ignored. As you can see, there is a margin between blue and red, and when we use information from the samples, it can improve the gene ontology enrichment analysis. Here, since it's negative logarithm, the higher, if it's higher, it's better. And finally, these are the top geo terms for this data. They are mostly related to nervous system naturally. And we can see that the top one is type 1 interferon response. This is classically attributed to viral infection, which is well-established, has a well-established connection to autism. And when we don't incorporate covariate information, we can see that this module, along some other important modules, gene ontology terms are missed. So the impact of covariate information can be significant sometimes. So to the conclusion, we developed a model that can automatically incorporate covariate information. It's directly applicable to count data from RNA sequencing. It automatically infers the number of factors, and it needs minimal intervention from the user unless there is a significant prior information that can be used for this model. Also, there are potential extensions to this model since people now are mostly using single-cell RNA sequencing technologies. We can add a zero inflation term to adapt it to single-cell data, which is more sparse compared to bulk RNA sequencing. And compared to network-based models, the one good property of network-based model is that we can directly model the correlation, positive or negative, between pairs of genes. For latent factor representation models, this is done using the latent factors. So we can have a more structured correlation on these latent factors to see how, for example, the expression of one factor is changed with respect to another one. So we can have, we can infer the correlation between groups of genes, not just consider them separately. So thank you for your attention. If you have any questions, I'll be happy.
Nice work. I was curious if you looked at enrichment of cis-regulatory elements in the modules, and how do you find new things, different things compared to the more conventional methods?
For Autism Data set?
Yeah, and also your other test data set.
So we looked at the gene ontology for all modules.
That I got, but the cis-regulatory elements, like motif
No,
elements.
we didn't.
That would be a nice.
We just take a look at the gene ontology.
So in terms of when you compare to WGCNA, did you compare to an approach where you first do a correction for the covariates as like a pre-processing
Yes.
step and then apply WGCNA? Or
That's right. So we have two conditions, treatment and control. For WGCNA, we applied them independently to each group. Then we applied the t-test. For the other method, DIF-COEX, it's an extension of WGCNA, which is kind of a heuristic way of adapting WGCNA to multiple conditions. So they do the differential expression first and then apply the network-based. So it's all considered, yeah.
Okay, thank you.
Can you comment on running time?
Since this is a Bayesian method, it can take several hours, but there are parallelization that can be used to, but it's not, it can be used for real time, but mostly 1 hour or so if you use parallelization.
Okay, let's thank all the speakers of this session. It's going to be the coffee break now, and we'll reconvene.